Okay, welcome everyone. Today we will have our second step seminars of the semester with Tayfun Özçelik, and he serves as the Dean of the Faculty of Sciences at Bilkent University. Thank you very much for being with us today. And if you want, you can start your presentation.
Okay. I need to share my screen, so could you please allow me to do so?
Okay.
You can.
So I'm guessing that you now see the screen.
Yes.
Excellent. Well, good evening. It's a pleasure to be with you today. I'm going to be talking about the genetic basis of obesity in humans. Actually, in a way, we also talk about the most important gift that our mother and father ever give to us. These are the two sets of 3 billion letters of DNA that make our genome. But like anything with 3 billion components, that gift is fragile. So sunlight, smoking, unhealthy eating, even spontaneous mistakes made by our cells all cause changes to our genome. So the most common kind of changes in our genome is in our DNA is a simple swapping of one letter or base, such as C to a G with a different letter, or a T or an A. In any day, the cell in our body will collectively accumulate billions of these single letter swaps, which are also called point mutations. Now that most of these point mutations are harmless, but every now and then a point mutation disrupts an important capability in a cell or causes a cell to misbehave in harmful ways. If that mutation were inherited from our parents or occurred early enough in our development, then the result would be that many of our cells contain that harmful mutation. And then we would be one of hundreds of millions of people with a genetic disease such as obesity or sickle cell anemia or progeria or muscular dystrophy or Tay-Sachs disease. So gravest genetic diseases caused by point mutations are especially frustrating because we often know the exact single letter change that causes the disease, and in theory, we could cure that disorder. So millions of people suffer from sickle cell anemia because they have a single A to T point mutation, both copies of their hemoglobin gene. You see here the red blood cells and the sickled cells as a result of a change from an A to a T. Or let's think about children with progeria. These children are born with a T at a single position in their genome, whereas we have a C with the, and that it would be our greatest goal to correct such a genetic change. So these are different types of diseases that we're talking about. And in fact, today I would like to focus on extreme forms of obesity. When we talk about these such diseases, actually, the first thing that one should concentrate on is the genomics era that we are living, and I would like to give you some background information on that. You know, there is the Mendelian inheritance in man database. Actually, this is now in the online version, so it is OMIM. I am guessing that the people who are listening to the seminar know about OMIM and entered to it and have seen different loci. Actually, this was started by a physician, Victor McCusick, who passed away some 14 years ago. Actually, he was a professor of internal medicine at Johns Hopkins University. And here you see the Mendelian inheritance in man, a catalog of all human mutations. And this is the first volume of it when it was first published in 1966, and then it was published in the form of a encyclopedia until 1998, and then it was discontinued. The print version was discontinued, and it all became an online database where it is continuously updated by the dedicated people. So here you see what McCusick did is in the early 50s and the 60s, he began describing different families with different genetic conditions, and these are pictures of Dr. McCusick, and these were compiled in this catalog. So actually, when we look at the numbers today, we know that there are about 50,000 human genes, and 25,000 of them are protein coding genes, and 25,000 of them are RNA genes. Well, when you look at this total number of 50,000 genes, the function of the gene is known for approximately 81% of them, and around 20% of them, the function of the gene is not really known. When you look at the diseases that are associated with these genes, we actually have now approximately 23, 24, 25% of them are associated with a human phenotype. A human phenotype meaning in most of the instances a specific disease. And about 60%, 57% of them is not associated with any disease at all. And actually, these diseases, single gene disorders, we also call them Mendelian diseases. Gregory Mendel was, you all know from your basic classes in genetics, is a monk who lived in Czech Republic in the 1800s. Actually, he was born in 1822 and he passed away in 1884. Mendel described the inheritance pattern, the laws of inheritance in studying peas, but what he discovered is applicable to all organisms, including humans, and that we call these single gene disorders Mendelian phenotypes. And medicine has been immensely successful in identifying these single gene disorders. In fact, when you look at from a global scale, from a vantage point, what you would see is that in the past, from 1990s to the beginning of 2000s, the Human Genome Project was done. Approximately 8% of the human genome was the remaining portion, and I think you all read it in the news a few days ago that the remaining 8% is now also solved. This 8% is like a single human chromosome, this content is. Of course, with so much information available, the age of bioinformatics next began. And then, of course, genome sciences flourished during the past decade, where the biology of the genomes and the biology of the diseases are now being solved. From 2020s onwards, of course, we have so much sequence information compiling. The big data analysis becomes really important, which eventually leads to developments in treatment. So in this background, actually, the single gene disorders that the research community has shown tremendous success, actually, they wanted to carry out, the research community wanted to carry out to the complex diseases. By complex diseases, what we mean is that the inheritance pattern is complex. Single gene disorders are also complex, but as far as the disease is concerned, but their inheritance pattern is so straightforward. This is not the case in complex disorders. So they are resistant to the identification of the associated disease genes. So International HapMap project, following the Human Genome Project, was initiated to tackle diseases like obesity, type 2 diabetes, different types of behavioral phenotypes like the psychiatric disorders also, and cardiovascular diseases, autoimmune diseases, Parkinson's, and Alzheimer's disease. In doing these experiments, actually, these gene identification studies, one of the starting points is the heritability index that I would like you to remember very well. What happens is that even if you don't have any information on the nature of the gene that is mutated, by looking at the heritability index through the analysis of pedigrees, you can decide whether that disease is amenable to gene identification. So these complex, let's say height is 80 to 90% heritable. Obesity is 70 to 80% heritable. Schizophrenia, a devastating psychiatric disease, we have 50 to 80% of heritability. ADHD, attention deficit hyperactivity disorder, that has both childhood and adult types. Well, actually, in ADHD, 60 to 70% of the affected individuals, they have a affected family member. Type 2 diabetes, about 40 to 60% of them are heritable. Major depression, another disease that we see quite frequently in the population. Well, here the heritability index goes to 30 to 40%. Breast cancer, you remember the genes for BRCA1 and BRCA2, these famous genes. Well, actually, when you look into people with breast cancer, approximately 20 to 25% of these individuals have a BRCA1 or BRCA2 mutation. In fact, when the consecutive studies were performed, taking into the analysis pipeline, all diagnosed cases of breast cancer in a particular clinic, approximately 20 to 25% were shown to be BRCA1 or BRCA2 carrier. This is also true for colon cancer, hereditary non-polyposis colorectal cancer, and APC, adenomatous polyposis coli. We have about 10 to 20%. Actually, when you look in general, different types of cancers, you see that nearly 10% of them are due to familial cases. Then there are diseases like Rett syndrome, where the heritability index is 0. If the heritability index is 0, then does it mean that that disease doesn't have a genetic basis? Well, the answer is no. Rett syndrome is a female mental retardation in which the mutated gene in males is lethal. Those males who inherit the Rett syndrome mutation, they die. And females, they have zero reproductive fitness, so they don't have children. So as a result, all cases are due to new mutations, and the heritability index consequently becomes zero. So from this list in general, we thought that obesity is a good candidate to study. And about now 10 years ago, we started our research. So starting from the 2012 or 14, coming on until 2020, the beginning of the pandemics, although obesity is such a disease with high heritability index, the genes, the causal genes were known in approximately 5% of the people. So 15q mutations, chromosome 15q associated with Prader-Willi syndrome, leptin receptor, melanocortin-4 receptor, pro-opiomelanocortin, PCSK1. These were the only known genes. So we thought obesity is a good candidate to study. So I would like to now introduce you to our research group. We have an international collaboration that involves Rockefeller University, Jeffrey Friedman, the discovery of the leptin hormone, and then from Rockefeller University, and then from Yale University, Kaya Bilguvar. Kaya Bilguvar is now in Turkey, back in Turkey, in Acıbadem University. We have Yuval Itan, who was at Rockefeller University when we started, but now is at the Mount Sinai Medical Center in New York. Myself, and then my postdoc, Emre Onat, who is now a faculty member at Acıbadem University. Caner Çağlar, a PhD student that from Bilkent University, who went to Rockefeller to study in Dr. Jeffrey Friedman's laboratory, and Junmin Choi, another postdoc at Rockefeller University, who is now a faculty member in Korea, and Shu Zheng, a PhD student who helped us in our analyses. And of course, my wife, İclal, who has been very instrumental in our connections with the families. So what our strategy in our research is to concentrate on extreme phenotypes, extreme meaning body mass index, a measure of obesity, people with a body mass index over 40, over 60. We concentrate on such individuals. You know, the normal range for body mass index is below 25. From 25 to 30 or so, one becomes overweight. From 30 to 35, obesity, and then extreme obesity when it is over 40 or 60. So we concentrate on people with body mass index of 60. And in the beginning, we only collected families that are consanguineous. Consanguinity means they are related with each other. But later on, when we looked at the composition of the families, we saw that anyone with a body mass index of over 40, majority of them are anyhow consanguineous. And then, of course, intrafamilial controls with a body mass index of below 25 was something that we wanted in our families. So what we did is we compared the genome sequences of people with low body mass index versus high body mass index. So our sampling strategy, I would like to share with you, is also a hallmark of our study. We did not wait for the patients to come to us. Instead, we have a team, including myself and Emre and a nurse, and we went to the families to sample them. Of course, through such family visits, one also has the chance to reach to more members of a given family. So here, for example, look, these are siblings, and look, one of them is obese, and the other sibling is thin. So this is an excellent opportunity to study the genomic differences. Or look at this family and look at this man who is relatively thinner than his brother who is so fat. Or as a matter of fact, look at this man also with an extreme form of obesity. And we sampled the families and then came to the laboratory as a team, and my students, you see them, and we extract the DNA from the samples and subject them to whole genome analysis. So we have families from all over Turkey. In fact, these are the provinces of the country, and the number of alleles coming from each province in Turkey. So we have 412, for example, families from or alleles from Ankara, or 80 from Istanbul, and almost all of the, not almost, in fact, all of the provinces, all of the cities are represented in our cohort to study the genetic basis of obesity. So let me give you a cohort description. We have a total of 4,296 genome sequences in our database. This happens to be the largest genome sequence database for the Turkish population, and 830 of them are whole genome sequences, and about 3,000 of them, 3,500 of them are exome sequences. So our obesity cohort consists of 1,152 families with 2,477 individuals, and we exome sequenced nearly 1,100 of them. And there are Turkish controls cohort consists of probands with neurological disorders and their healthy relatives. So 77% of our probands were self-reported the disease as onset as childhood. Self-reported consanguinity and endogamy is highly observed among parents of the probands, which is calculated as around 40% by homozygosity by descent analysis. So in order to assess the quality of our data, we evaluated the sequencing data at each level of the pipeline using sample level fast QC analysis, base level BQSR, and variant level BQSR analysis. We excluded low-qual samples and variants dependent on the analysis pipeline. In order to assess the quality of our data, we evaluated the sequence data at each level of the pipeline using sample level fast QC analysis, base level BQSR, and variant level BQSR analysis. We excluded low quality samples and variants dependent on the analysis pipeline. So for case control collapsing gene association tests, we examined population structure using principal component analysis. of the Turkish individuals and excluded the outliers. So you see here the results of the principal component analysis, and actually I would like to show you a blow up of this region here. If you concentrate, the red color is the Turkish population. Here you can see it much better. The green happens to be the Balkans, and then the blue is the Europeans, the purple is the Middle Easterns. Actually, if you look at this scale, Africa is out here, and South Asia is here, and other populations are Middle Eastern populations are up here, and we have actually and this is the Balkans and the Caucasus regions, and you see that Turkey, like our geographical location, which is like a bridge between Asia and Europe, we have overlap with the European populations, and we are intermixed with the Balkans and the Caucasus regions. So, we have overlapping with different populations. We used King tool to infer relatedness among Turkish cases and controls. Any two individuals that are second degree kinship coefficient of 0.0625, relatives relatives were removed before the analysis. A total of 3,362 samples remained after filtration according to quality metrics and relatedness. For family-based collapsing analysis, we generated kinship matrix using MMAX software by taking into account pedigree structures of our families and the genotype data of individuals. MMAX simultaneously creates a correlation between phenotypes of samples by a combined linear mixed model of population stratification and relatedness. So we selected the exome parts of the whole genome data and combined with the that of the exome set and evaluated the data by burden and scatter collapsing tests using the synonymous variants and singletons at different minor allele frequency cutoffs. These are 10 to the minus 3, 10 to the minus 4, and 10 to the minus 5. There were no inflated or deflated variants in the analysis. This scatter here, you see that it is on the single line, or here it is on the single line, or here also very close to this single line, showing that this data is reliable, and that gene discovery based that on such data would be possible as an indication. We also filtered variants based on transition and transversion ratios. You know, there are the transition and transversion types of genetic changes of VQSR, genotype quality depth and mapping quality. Following a variant filtration process to identify high quality variants, we observed 1.25 million variants for downstream analysis. We first analyzed obesity-related genes using two different approaches, a gene-based segregation approach and a gene-based burden analysis for identifying rare variants. Well, actually, I told you that up until 2020, there were only a handful of genes that were known to be associated with obesity. Starting from 2021, a big difference emerged because this is because of the Genomics England and the US databases that become now available with the genes that are associated with obesity. Now, there are a total of 113 genes that have been reported in Genomics England, ClinVar, clinical variation, it becomes from OMIM, Online Mendelian Inheritance in Man, and Human Gene Mutation Database. And this is a Venn diagram. Let's say these genes shown here are reported only in Human Gene Mutation Database, whereas these genes, they overlap. They are both in ClinVar and in HGMD. So here, if you come here, these are the genes that are represented in all 4 databases. So in fact, and the ones that are in red are the genes for which the animal model is also obesity. By animal model, I mean the knockout phenotype of the mouse. So there are high penetrance OB genes like leptin, leptin receptor, POMC, MC4R, PCSK1, BDNF, and so on. There are syndromic obesity genes. These syndromic obesity genes, some alleles of that mutation in that gene result in a distinct syndrome like Alström or Cohen syndrome or Borjeson-Forstmann-Lehmann syndrome or Albright hereditary osteodystrophy. And then some other mutations of the same gene, a different domain of that gene, the syndromic features may be less pronounced and the obesity becomes the cardinal feature. Of course, all of these syndromes are in fact also in their syndromic form associated with obesity. And of course, digestive hormones like ghrelin or cholecystokinin or gastric inhibitory polypeptide or glucagon like peptide and so on are also associated with a tendency to obesity. And this is what we have analyzed in our families. And in a gene burden analysis, we classified variants in 4 models. These are the recessive model, loss of function model, dominant model, and ClinVar model. And the dominant model was divided in strict and broad models. And here you see a volcano plot. This volcano plot shows the gene burden, gene-based burden analysis results. And here you have the most significant ones like ENPP, MC4R, LRP2, and BBS2, Bardet-Biedl syndrome number 2 genes are implicated. So here is a statistical distribution of the candidates. I know that this is a slide that is difficult to read, but it gives you a general idea of the distribution of the candidates. And the blue bars indicate the number of variants, and the orange bars indicate the number of families for each gene. And actually, let me show you this with a enlarged portion of this figure here. We identified that of the 799 unrelated families that have been genome sequenced, 244 of them. This corresponds to nearly 30%. This is a large number, 30%. They have a mutation in one of the obesity-associated genes. So of the 113 obesity-associated genes, we found that 63 genes, these are harboring mutations in our cohort. So there is the gene-based burden analysis and gene-based segregation analysis here are shown, and the number of families under different models are depicted in the figure. This is again a difficult figure to follow, but it gives you a general idea. Here, the red colors are recessive and the blue are dominant, and this shows you the family-based cluster analysis. The y-axis includes the candidate genes. These are the candidate genes, and the x-axis includes the families. So for a particular gene, you have so many numbers of families that are represented. So when we analyze clustering of the mutations to the families, we observed that more than one gene is implicated in 28 of the families, which happens to be around 10%. So actually, this is a more digestible, I think, figure. And here you see the combined summary table of obesity-related analysis, and these are the familial OB genes. So we have variants in MC4R, leptin receptor, leptin, and PCSK1, and these are the different percentages of the families. So 11 different mutations for MC4R in 26 families. The OB-related and mouse knockout OB genes, well, here are the names of these genes like ADCY3 and CPE and PPAR gamma and so on. And we have so many number of variants and so many number of families are implicated. With respect to OB-related genes, also we have the order here with the gene name and the number of variants and the number of families associated with these mutations. So here you have the general outline of these. So, and continues with the same pattern. There are single variants and single families for many of like FTO and so on. So actually, as a summary slide, the obesity-related genes, this is another Venn diagram, and we have 21 variants in 44 families. This is about 5.5% of the highly penetrant OB genes, and the remaining 113 genes distributed into different phenotypes. Now, actually, again, this shows that nearly 30%, more than 30% of the families, we can identify a genetic basis. This is a high number. This is an important observation. So when we look at the pedigrees, since families are so important for our analysis, I would like to show you some pedigrees. This is a family tree, and you have a let's say, a leptin mutant with 2 homozygotes, and then there are the heterozygotes or wild-type individuals, and the weight of these individuals, when it is a black symbol, then this means that these are really obese people, and then with somehow obese people are gray, and we have these families. Leptin receptor is another gene that we concentrated on and identified these families with their mutations. Let's say the home here means that these are homozygotes, and these are heterozygote individuals, heterozygotes, or and so on. Since obesity is a really prevalent phenotype, we see phenocopies. Phenocopy means that in the same family, you have individuals who do not have the mutation. Look at this one, for example, wild type. She is a phenocopy of her sister, which complicates the genetic analysis. MC4R, for example, here are the families with different types of mutations that are depicted, or PCSK1 mutations or POMC mutations in the families. Well, I would like to now concentrate on an important observation that came from our studies in these families. Heterozygotes also display a phenotype in their 4th to 2nd decades of their lives. This means that, some diseases like familial hypercholesterolemia associated with LDL receptor mutations, low density lipoprotein mutations, these families have the phenotype as expressing the phenotype during childhood in homozygotes and then in heterozygotes during their 40s. So this is exactly the same for obesity and that leptin mutation carriers. Here we have one such family. Now this girl and this boy here are the homozygotes and these are the siblings. Look at their mother when she is in her nearly 40s. She is 39 years old or 40 years old now, even more than 40 years old. Now she has a body mass index of 37. Or look at the father at 33. Or look at the upper generation. Here we have people with body mass index of 46, or here we have she is 52 years old and she has a body mass index of 49. This has implications for genetic counseling. Well, when you look at the zygosity ratios, when a homozygote is 1 in 2,000, this means that heterozygotes would be 1 in 20 to 25. So there is a 100-fold increase. This means that rare phenotype, this is the Hardy-Weinberg equilibrium. This is the basics of any genetic analysis, Hardy-Weinberg. Well, this means that leptin, for example, such a rare disease as we know, there are a handful of families. Well, the heterozygotes are not so little in their numbers, and that serum leptin levels, when you look at them, you can also see the associated results comparable with the genotype data. So actually now I am coming to the final portion of my talk. Actually, you're going to ask me, what about novel genes? Do you discover any new genes? Well, indeed, we are studying them, and I would like to show you some more slides about this. For disease gene discovery, we use two main approaches: case control analysis using gene-based burden test and family-based analysis using scatter test with a max kinship. And because of time constraints, I cannot show all the analysis, but here is an example of gene-based binomial burden analysis. This is our filtering and prioritization analysis workflow, and test using variance with minor allele frequency cutoff of 10 to the -4, and we use different filtering approaches for missense variance, and we have the case control analysis with the scatter filtration, and we are looking at the popping up genes. So here is a list of our best candidates. These are the candidates were combined from many analyses done. P-values are the highest P's from each model, and the numbers are related with that P. As a total, we have 271 variants in 36 genes, and together with our collaborators, we are working on prioritizing these novel gene variants which we hope to identify new obesity genes. And in this study, we also had a novel approach. This is the PHEVAS analysis, phenome-wide association studies. So we conducted two independent PHEVAS analyses, top phenotypes for each candidate gene and top genes for OB-related phenotypes. And we came up with interesting results. So you see that we found significant association between obesity-related phenotypes and one of our candidate genes. Here it is at the very top. Because of the ongoing studies, I do not want to go more into detail of this candidate, but this candidate actually, when we look at the variant in the whole phenotypes, we saw that it is highly associated with individuals with a body mass index of 30 to 40, so classified as obese. Actually, these PHEVAS analyses were conducted using the Mount Sinai population database, which is a restricted database, but because of our collaborative study is with the people who have access to the data, we are capable of performing these phenotype studies. So actually, this is a very encouraging result. And what you do when you get such an encouraging result is that you go ahead and analyze more genes. And we have here those that are crossed out are the genes that looked very promising at the beginning, but through our segregation analysis in the families, they turned out to be not segregating. So since segregation did not support, these genes were excluded. However, the remaining genes, we have interesting results. So here is one such segregation analysis with our candidate gene. Here is the candidate gene with the mutations and the localization of these mutations on the structure of the gene. You have the exon intron structure, and then these are the individual mutations that are found in this gene. So this is the 5' regulatory region, and this is the coding part of the gene. And here is my final slide. This is, you see, there is a segregation, obese person, heterozygote, obese, heterozygote, and this is now 6 years old, too young to be evaluated, but still we have another family member who is in the obese range with a heterozygote phenotype. Or look at this family, heterozygote, heterozygote, heterozygote, heterozygote, and let's say this non-obese individual is wild type. So to sum up what I have described to you, we actually attempted the molecular dissection of a complex human phenotype, obesity, and through genome sequencing during the past 10 years, we compiled the largest Turkish genome database and studied more than 1,000 families with extreme forms of obesity. And we first looked at the known OB genes, and we have now results that show nearly 30% of the families harbor mutations in these obesity-associated genes, and that when we look into the novel gene discovery aspect of our cohort, we have some pretty strong candidates. One of these candidates that I did not mention here is associated with our taste buds, the way we taste a certain. There are basic tastes like tasting for sweet, tasting for sour, tasting for umami, tasting for bitter, and in one of these specific receptors for our taste, there is a mutation. There are several mutations in several of our families, and that right now the further studies like introducing these mutations in model organisms is ongoing. I think this concludes my talk for today. If you have questions, I'd be happy to answer them. Thank you very much for your attention. Yes.
Thank you. Is there anyone would you like to ask a question?
Are you all molecular biology and genetics students?
Not now, but I will choose this program.
Oh, of course. You are a first-year student or a second year?
Yes, I'm in prep class right now.
Okay. So some of my presentation, some portions of my presentation was a bit too, I think, specific. But anyhow, this is how the terminology we use.
Yes, thank you. Probably people are breaking their fasting right now. That's why they don't have any questions.
Yes.
But thank you for coming, and thanks a lot for this good presentation.
You're very welcome. Very welcome. So I wish you all a good semester. And study your lessons and have excellent grades. I hope I also inspired some of you to pursue a career in molecular